Drought is not just a lack of water. For a plant, it's a chain reaction. As soil dries, cells lose turgor, growth slows, stomata close to prevent water loss, carbon dioxide can't enter as easily, photosynthesis drops, and damaging reactive oxygen molecules build up. Reproduction and yield are often the first things to fail. So drought tolerance is not controlled by a single gene. It's a network of traits that trade survival against growth. This is where CRISPR comes in. CRISPR is a gene editing tool that makes precise changes to a plant's own DNA. A short guide RNA brings the Cas enzyme to a specific DNA sequence, the DNA is cut or modified, and the plant repairs it. In crops, the most common result is a small mutation that knocks out a gene or subtly changes how strongly it is expressed. Usually, no foreign gene is added. Scientists use CRISPR to edit genes that control how plants manage water and stress. One major target is stomata. Stomata balance water loss against carbon intake and are controlled by the drought hormone abscisic acid, or ABA. By knocking out negative regulators in ABA signaling, or by tuning gene expression in guard cells, plants can close stomata faster during drought, conserving water without fully shutting down photosynthesis. Another strategy focuses on cell survival. Under drought, plants accumulate osmoprotectants like proline and certain sugars that stabilize proteins in membranes. CRISPR can remove repressors of these pathways or adjust their regulation so protective molecules build up only during stress, limiting yield penalties when water is abundant. CRISPR is also used to modify stress response transcription factors, such as DREB or NAC proteins. These act high in regulatory networks, switching on dozens of drought response genes at once. Instead of permanently activating them, researchers often edit promoters so these factors turn on mainly during drought, avoiding stunted growth. Roots are another key target. Deeper or more angled roots can access water stored lower in the soil. By editing genes involved in root growth and hormone signaling, CRISPR can shift root architecture toward better water uptake, often with root-specific effects. A crucial agricultural goal is yield stability. Some stress hormones, like ethylene, reduce growth under drought. In maize, CRISPR has been used to fine-tune expression of Argos-8, a negative regulator of ethylene response, improving grain yield under dry field conditions by adjusting an existing gene rather than adding a new one. The workflow is conceptually simple. Researchers identify candidate genes using genetics and drought experiments, choose whether to knock out or tune a gene, edit plant cells with CRISPR, regenerate whole plants, confirm the exact DNA changes, and then test performance in controlled drought and real field trials. Many edits fail and trade-offs are common. The big picture is this. CRISPR helps engineer drought-tolerant crops by optimizing decisions plants already make: when to lose water, how to protect cells, where to grow roots, and how much growth to sacrifice. It's powerful, precise, and fast, but drought tolerance still comes from careful fine-tuning of complex biology, not from flipping a single genetic switch.